Okay, so Bob has been grinding the tissue.
So we've just got a little bit of tissue. We haven't actually used liquid nitrogen or dry ice for this experiment because we haven't got any. But what we do is we just simply grind the material to try and make it into, crush the cell, well, crush that material as much as possible. Now we're doing this experiment in duplicates, so we're actually using the same material. We're actually doing essentially 2 reps of the samples. So we're actually physically grinding this as much as possible.
Yeah.
And then what we do now is we add 500 microliters of shorty buffer. So what we have to do is we have to actually set our pipette to 500 microliters, and then we transfer some of this shorty buffer to each of our reactions. And what we do is we still continue to grind that material in our shorty buffer. And so the shorty buffer, what it does is it actually will rupture the cells, the plant cells, to allow that DNA to come out of those cells. So we just continue grinding as much as possible. Now the beauty of PCR, which is a technique that we're going to do later on, is you don't actually need a large amount of DNA material in order to actually amplify it. So what we're doing now is we're just physically grinding up that plant tissue as much as possible, as much as practically possible. Now what we've done is, once we've finished grinding that up, we put our dirty pestles into our waste tips beaker. And the reason we do that is we don't want to put them back into a clean pestles beaker, so therefore we prevent or reduce our risk of cross-contamination. Now what we're going to do is we're going to go and centrifuge these samples for 5 minutes at maximum speed at 4 degrees. So what we do is we'll come over here. Now because this is a mock demonstration, we won't actually need to run this centrifuge at 4 degrees. We can just run it at room temperature. But if you were in a research laboratory and you would actually do this, for a real experiment, you would actually do that at 4 degrees to reduce the amount of damage or degradation of the DNA sample. Now it's important to note when you actually put your microcentrifuge tubes, or your Eppendorf tubes we call them, but microcentrifuge tubes, the 1.5 microcentrifuge tubes, you need to put them directly across the axis of the rotor of this centrifuge to make sure that when it spins, it actually spins balanced, or centrifuge is balanced. We then place it there. We crank up the speed to the maximum it can go. In this case, 13,200 revs per minute. And we're going to spin this, or allow this centrifuge to go for 5 minutes. It's important to make sure that we put the lid on top. And just another trick of the trade is to actually, where the hinge, where the lid goes onto the microcentrifuge tube, you just actually point it outwards. So you'll actually get a nice pellet formed at the bottom of this microcentrifuge tube, and you'll actually know where that pellet is formed based on where the direction of that hinge is. If you point the hinge out, the pellet will be on one particular side where the hinge is. Okay? And now we start. Right, let's go to maximum speed, 13,200, and you can hear there's no vibration, the centrifuge is running well, or evenly.
So you've balanced it properly, Bob?
So the centrifuge is running balanced. The samples in the centrifuge are balanced with one another.